Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. I warmly welcome all of you to my channel Science Champion. Today in this video we will talk about enzymes. What are enzymes? Enzymes are complex proteins that speed up the rate of chemical reactions. For example, if any reaction is occurring in 4 hours, so by adding enzymes it can be done in 2 hours. It actually speeds up the reaction. Enzymes remain unchanged at the end of chemical reactions. For example, if we talk about digestive enzymes. So chemical reaction occurs when complex food molecules are broken down into smaller food molecules. So these are called digestive enzymes. Enzymes help to speed up the rate of such chemical reactions. As a result, digestion occurs faster. The enzymes involved in the digestion of food are called digestive enzymes. There are some key terms related to enzymes like catalyst, enzyme, substrate, active site. Let's understand what is catalyst. A substance that speeds up the chemical reaction without being changed. It is actually the enzyme. Like enzyme is a biological catalyst. Substrate. It can be the food molecule because it is the reactant molecule that an enzyme works on, or anything for which we require the enzyme. Active site, the part of the enzyme where the substrate binds. Now just focus that how these key terms are involved in the working of enzymes. Look, this is an enzyme, the first one. Then there is a substrate. When this substrate binds, it makes the enzyme substrate complex in the next one. Now after that the reaction will occur and substrate will convert it into products, and you can see that the enzyme is unchanged at the end of the reaction. Another diagram in front of you. It is also called the lock and key model because it was designed by a scientist, Emil Fischer, who have suggested that it is like a key. Like the key is specific for a lock. Similarly, every enzyme is specific for the substrate. Look at the enzyme. It has an active site, and that active site is specific for the substrate. Only that substrate can bind with this active site for which has been designed. When it will bind, it will form the enzyme substrate complex. And after the reaction proceeds, the product will be there and the enzymes remain unchanged. Now we will talk about activation energy. Like what is activation energy? Basically, enzymes do what? Enzymes lower the activation energy of a reaction. It is the required amount of energy needed for a reaction to occur. They do this by binding to a substrate and holding it in a way that allows the reaction to happen more efficiently. For example, look at this picture carefully. On x-axis, we have taken the reaction progress, that how the reaction is occurring, what is the progress of the reaction. And on y-axis, there is the required energy for the reaction is there, that is basically the activation energy for the reaction to be started. First, focus on the purple line. This is the purple line. As you can see, the activation energy of this uncatalyzed means this is the time when the reaction is occurring without the enzyme. So it is more, and the reaction will start in more time. It will take more time to progress. Now, as you can see in this green line, this is the activation energy of catalyzed reaction, like when we have added up some enzymes. So when we add enzymes in it, so enzymes will lower down the activation energy needed for the reaction to occur, and the reaction occurs speedily. It will take less time to start and to proceed. It will proceed quickly. It occurs speedily. The reaction progressing quickly. There are some factors that can affect the activity of enzymes, like temperature, pH, and concentrations. These are the factors over which enzymes work best, and they are called optimum conditions for the working of enzymes. Enzymes work best when a specific temperature and pH ranges, and suboptimum conditions can cause an enzyme to lose its ability to bind to a substrate. Like there are various factors, there are various optimum conditions for the working of enzyme. The first one, temperature, then pH, then enzyme concentration, and also the substrate concentration. Temperature, when we talk about the temperature, so when we increase the temperature, it generally speed up the reaction. More the temperature, more speed up the reaction. And lowering the temperature slows down a reaction. It has a direct relation with the temperature. However, remember one thing, at extreme high temperature can cause an enzyme to lose its shape, it denature it and stop working. So the temperature must not be too much high, it should be optimum temperature over which the enzyme works best. Then we have pH. Each enzyme has an optimum pH range. Changing the pH outside of this range will slow enzyme activity. Extreme pH value can cause enzymes to denature. Again, the same thing, when you increase the temperature, the enzymes will denature. When you increase the pH, when the pH is extreme, so the enzymes will denature. They also work at optimum temperature. Similarly, they also work at optimum pH. Enzyme concentration. Increasing enzyme concentration will speed up the reaction as long as there is a substrate available to bind it. When you have a reaction, you have substrate, you are adding up enzyme, more the enzymes, more it will speed up the reaction. Similarly, you have substrate concentration. Increasing substrate concentration also increases the rate of reaction to a certain point. Certain points means the substrate must be available. It is not like that you are adding the substrate more and more and more and the enzymes are there, it is speed up, and until and unless there is no enzyme and the substrate are more, so it will not speed up the reaction. So substrate concentration, increasing substrate concentration also increases the rate of reaction to a certain point in the presence of enzyme. Here you can see the applications of enzymes. We have various applications of enzymes. Enzymes are useful for the environment as they take part in waste degradation, as they take part in detection of toxic pollutants. Similarly, we have various enzymes that are helpful in industry. We have various enzymes that are helpful in agriculture. So these are the applications of enzymes, that where we are using enzymes. We use enzymes in leather tanning, we use enzymes in paper and pulp production. We use enzymes for the starch and the fuel. Chemical and pharmaceutical companies are also using enzymes. We have enzymes also present in the detergent. We have enzymes in the food and beverages, textile, protein hydrolysis. We use enzymes for the animal feed from agricultural waste. There are various applications of enzymes in our environment. Like you can see, they are used as biocatalyst, biotech, diagnostic point of view. We are also using enzymes in food and beverages, in animal feed, in pharmaceuticals, in biofuels. So we are using enzymes in our daily life. So this was all about my video. Jazakallahu khaira. Thank you so much for watching my video, and don't forget to subscribe my channel.